did many crosses in Garden Pig clan to find out the law of inheritance. Now, here he wanted to know whether sex is there any influence in the inheritance of trait. That's why he did one cross, what is called reciprocal cross. What do you mean by reciprocal cross? Here he did two crosses. In one cross, he took the tall plants and pollinated the tall plants by taking the pollen grain from the dwarf plant. That means, in that cross, the pollen grains from the dwarf plant are transferred to the tall plants. So we can consider, now this tall plant is only with the pistil, hence it is considered as a female plant. As the pollen grains are taken from, that is a dwarf plant, and this is considered as a male plant. So in that cross, he observed and obtained all the plants were tall. This is one cross. In the next cross, he reversed the sex. Now, what is the meaning for that one? So instead of taking pollen grains from the dwarf plant, he had taken pollen grains from the tall plant and transferred the pollen grains into the stigma of the dwarf plant. Now here also, the result will be the same. The reason for that one, either the pollen from the tall plant or from the dwarf plant, the result will be the same. That means the tall plant is acting as a dominant character. Now, here the inheritance depends on the quality of the genes, either dominancy or recessiveness. As per the law of dominance, only the dominant character will be expressed in F1 generation. Now, based on this experiment, so what is reciprocal cross? So the mating, what we have, the reciprocal cross I will represent in a symbol V. Two crosses indeed in both the ways. So matings of one cross we have tall, he considered as a female, and then dwarf, he considered as a male. This is one cross. So matings of tall, that is a female, just I'm using the board because the pollen grains are transferred from that dwarf plant to the tall plant. In another cross, and this is another cross, that is tall. From the tall plants, the pollen grains are transferred to the stigma of dwarf. Hence it is considered as a female plant. This is called sex reversal, changing the sex. So here the sex is reversed. So the mating of tall plant with the dwarf plant, that is nothing but the pollen grain from the dwarf is transferred to the tall. The second one, mating of tall plant with the dwarf, here the pollen grains are transferred from the tall plant to the dwarf plant. Now here you see that one, the matings occur in both the ways. The matings occur in both the ways. Such matings occur in both the ways are called reciprocal crosses. The matings which occur in both the ways are called reciprocal crosses. In both the crosses, the result will be the same. In both the crosses, the result will be the same. In both the crosses, the result will be the same. That is a tall plants. So from these in such crosses, the trait inheritance is not sex dependent, or the monohybrid processes inheritance, or in monohybrid processes, the inheritance of traits is not sex dependent. That was a conclusion made by Mendel. That's why he did what is called the reciprocal cross, to show that one, the sex has no role in the inheritance of characters in his analysis. According to the Mendel's reciprocal cross, he concluded that the trait is not sex dependent. Sex has no role in the inheritance of traits. Now we have to go for the next one, back cross. What is back cross? Crossing of F1 hybrid. Crossing of F1 hybrid. Either with the dominant parent, or with the recessive parent. with the recessive parent. This is called backcross. Crossing of F1 hybrid individual either with the dominant parent or with the recessive parent is called actually backcross. So crossing of F1 hybrid either of the parents, crossing of F1 hybrid with either of the parents, we can say shortly is called as a backcross. Now what are the types of backcrosses? One backcross is called dominant backcross. One backcross is called dominant backcross. What is dominant backcross? Now here we are crossing F1 hybrid. Crossing F1 hybrid with a dominant parent. With a dominant parent. The crossing of F1 hybrid with a homozygous dominant parent. We can say, because we have heterozygous also available, homozygous dominant parent. So the F1 hybrid is seen normally a heterozygous individual. So it's being crossed with a homozygous dominant parent. What is the result? In the case of such dominant backcrosses, we have received all the individuals term. We have received all the individuals term. And this is called what is known as dominant backcross. Now this dominant backcross is of no use to find out the genotype of the F1 because we are getting the same result while making a cross between a homozygous dom and what is called a homozygous, that is dove. In these cases also we are getting odd term. So there is no difference between these two. We are getting the same result either between homozygous dom and homozygous what is called dove, or between F1 hybrid and homozygous dominant parent. The result would be the same. So it is not used to find out the genotype of the parent, that is a dominant parent involved. The main purpose of backcross is to find out the nature of the dominant parent, whether the dominant parent is homozygous dominant or heterozygous dominant. But in the case of this dominant backcross, as it is similar to what is called the normal cross, a monohybrid cross, a cross between purebred tall and purebred blow, we are getting the result all tall in the F1 generation. Here too we are getting the result F1 generation tall. So we can say F1 or F2 we can say. So we can say this dominant backcross is not used to find out the genotype of the parents, particularly the dominant parent. So we have to go for another what is called cross. That is called recessive backcross. Second type of backcross is recessive backcross. What is recessive backcross? In recessive backcross, the F1 hybrid tall is crossed with homozygous recessive parent. Suppose you are taking the genotype. This is the genotype. Now in the case of such crosses, we are getting normally here two different types of gametes. Here only one type of gamete. So the probability would be 1 into 2. Two types of individuals would be formed. Now in the case of dominant backcross, there is no appearance of recessive character. The character appeared is only the dominant. But in the case of recessive backcross, we have the appearance of recessive character. Now here if we make a cross, you see that one, both tall and low characters appear equally in equal proportion of 50% each. So this is 50% tall and 50% low. So in a recessive backcross, there is an appearance of recessive character. That's why normally we are taking this recessive backcross. Now this cross is used to find out what is called actually the heterozygosity of the tall parent. Whether the tall parent is homozygous or heterozygous. If it is homozygous, we can get all the plants are tall F1 generation. If the F1 hybrid is normally a heterozygous, when it is crossed with a homozygous recessive, then will give both the tall and dwarf glands in the ratio that is 1:1. So, if you are getting the ratio in a hybrid cross of 1:1, it shows that now the dominant parent involved is heterozygous. Only when the heterozygous parent is involved in that cross, then you have 1:1 ratio. But in the case of previous one, there you have the dominant parent is involved. So you are getting all the plants are tall. That is called the dominant back cross. So in dominant back cross, we are crossing F1 hybrid with what is called homozygous dominant parent. In such cases, we are getting the results all tall. But in the case of recessive back cross, we have two different types of plants in the ratio of 1:1. If in a hybrid cross, the ratio obtained is 1:1, then definitely we can say the dominant parent involved in that cross is heterozygous. We are getting 1:1 ratio only when the dominant parent is heterozygous. As this cross is used to find out the nature of the dominant parent, whether it is homozygous or heterozygous, it is also called test cross. It is also called test cross. So now we are doing the test cross for monohybrid cross, hence called monohybrid test cross. So test cross is the one which is a recessive back cross in which heterozygous or F1 hybrid is crossed with the recessive parent, leading to the formation of dominant and recessive characters in the ratio of 1:1. They have showed that the individual, the dominant parent is heterozygous, and we can conclude this test is used to find out the nature of the dominant parent, hence called test cross. What is test cross? It is one of the back crosses where crossing of an individual with unknown genotype with a homozygous recessive parent or homozygous recessive individual. This is called test cross. I mentioned already the recessive back cross is called test cross. The reason for that one, it is used to find out the nature of the dominant parent, whether it is heterozygous or homozygous for that character. Now, as per the book, if you are taking the recessive back cross or simply the test cross, one type of test cross is heterozygous tall test cross. Heterozygous tall test cross, in which the heterozygous parent is tall. The heterozygous parent is tall. Actually, I am taking the tall plant. The tall character is heterozygous, or we can say the parent is heterozygous, hence called heterozygous tall test cross. In heterozygous tall test cross, a heterozygous tall plant, this is unknown. We don't know the genotype of an organism. We are simply taking a dominant parent. It is being crossed with a homozygous dwarf or recessive parent. Now we see the possible parental phenotypes, heterozygous tall. We didn't know that one, but we have written. The result would be, that is what we are getting after. So heterozygous tall and homozygous dwarf. Now this is heterozygous. Now this heterozygous tall produces two different types of gametes. 50% with a capital T and 50% with a small t. That is dominant allele and recessive allele. Whereas a recessive parent produces only one type of actual gamete with the recessive allele. Now, as we have two different types of gametes on one side and only one on the other side, 1 into 2, the probability, the possibility of individuals with the probability in the generation is what we have actually 1:1. That is 50% tall and 50% tall. So the genotypes, these are the gametes formed. The offsprings of F1, as we are taking these individuals as the parental combinations or parental types, not parental combination, the parental types. We are getting what is called the offsprings of F1. That is genotype, one heterozygous tall, another one homozygous dwarf. Now what is the genotypic ratio? As we have 50-50 probability, so 1:1. Now if you are taking the phenotype, so 50% of the individuals are tall and 50% of the individuals are tall. So also we have the phenotypic ratio 1:1. So in the case of test cross, if the individuals involved are heterozygous dominant and homozygous recessive, then we are getting the ratio of 1:1. And that is the result what we obtain when the tall parent is heterozygous. So in this one, you have observed both the phenotypic and genotypic ratio will be the same. In both the cases, 1:1 ratio. That is, both the phenotypic and genotypic ratio will be the same. So if in a hybrid cross, if you are getting the ratio 1:1, then we can say it is an example for heterozygous test cross. Okay, heterozygous tall test cross, ascended test cross. Now actually, what is the importance of the test cross? The test cross is used to find out whether the parent, the dominant parent involved is homozygous dominant or heterozygous dominant. So getting the results of 1:1, definitely one can say the dominant parent involved is heterozygous dominant. Okay, so that is about what I have the test cross. So normally mental dig, the experiment want to know the genotypes. First, he obtained what we have heterozygous parent while making a cross between a pure tall and pure dwarf parents. Now he obtained all individuals F1 tall. This is F1. Now he crossed actually two F1 individuals and obtained 3:1 ratio F2. This is what we have the dwarf. So 3 tall, 1 dwarf will be produced. He crossed the dwarf varieties to get the results in F2 and F3. In all the F2 and F3 generations, while there is the dwarf parents were crossed of the F1, he got only the dwarf. Hence he concluded that dwarf character is homozygous recessive. So on to actually predict the genotypes of the F1 dominant parent. He wanted to cross actually, so he didn't know, he didn't predict the genotypes of the tall parent, whether it is that is heterozygous or homozygous. Want to predict the genotypes of the tall plant, obtained the F1 generation. He crossed this heterozygous tall, I am using the word heterozygous, simply hybrid tall with homozygous dwarf parent. And this cross is called actually test cross. Crossing of a dominant parent, unknown genotype, with a recessive parent is called test cross. So in that test cross, we obtained the following ratios. So the main purpose of test cross, particularly the heterozygous tall test cross, to know whether the dominant parent is heterozygous or homozygous, that obtain the F1 generation. So that is the conclusion. I will represent the checkerboard also after finishing if another cross, if homozygous tall test cross. Previous cross, otherwise on homozygous, otherwise on heterozygous tall test cross. It is used. Normally, it can be easily analyzed. The progenies and their genotypes and phenotypes. Now, in the case of homozygous tall test cross, we are taking the homozygotes, actually the tall parent homozygous, a true breeding one. We are not taking heterozygous. So we are crossing what is called homozygous tall with a homozygous recessive. That is a dwarf parent. Now here it is more or less similar to the monohybrid cross. As we are using homozygous tall, this is called homozygous tall test cross. Though it is called test cross, it is not analyzed. It is not used to analyze the genotype of what we have the progenies. That's why it's not useful in genetics to find out the nature of the dominant parent. Anyway, we have to go as per the book. So here we are taking homozygous tall parent, crossing the homozygous tall parent with homozygous dropper. The gametes produced by the tall parents are similar, each with a dominant allele. Similarly, the gametes produced by the homozygous recessive parent similar, having the recessive allele. So the random union of these two gametes result in the production of a progeny. Now all the progenies resulted from that cross have the genotype heterozygous condition. So this is heterozygous tone and that is heterozygous tone. But the result I can say all the plants appeared or tone. So in homozygous tone test cross, the resultant products are all tone. But in the case of heterozygous tone test cross, the resultant products are in 1:1 ratio of dominant and recessive. We can express both the heterozygous tone test cross and homozygous tone test cross in a Punnett square. So the test cross is now used clearly to identify and predict the genotypes and the phenotypes of the offspring. Now here I am taking the Punnett square. As we have the probability of 4 individuals, I am taking what is called the male gametes with the recessive allele on one side, the female gametes with the dominant allele and recessive allele, because we are taking the parent hybrid or heterozygous. Now what are the results obtained here? Now simple what's called the probability crossing of these two results in heterozygous tone. Here also we are getting heterozygous tone. And here we are getting what's called homozygous block and homozygous block, because the gametes involved have only the recessive allele. Now out of the 4 individuals, 2 individuals are heterozygous tone and 2 individuals are homozygous block. So the phenotypic ratio is 1:1. So out of the 4 individuals, 50% are tall and 50% are tall. So this is the actual test cross to find out the genotype. Okay, so test cross, you know, is very important to find out whether an individual is homozygous or heterozygous for a dominant character. If it is a heterozygous dominant, then the result would be 1:1 ratio. So if in a hybrid cross, the phenotypic ratio obtained is 1:1, then we can say the dominant parent involved is heterozygous dominant. Now let's go for the homozygous tall test cross. Homozygous tall test. So this is a heterozygous tall test cross and this one is a homozygous tall test cross, because we are taking the tall plant homozygous. Now on one side we are taking the female gametes, there is both, there is dominant letter, the dominant allele. On the other side we are taking the male gametes both with what is called the recessive alleles. Now as a result of the union, we are getting all the individuals are heterozygous tone. So this is what's called, otherwise called dominant back cross. We are crossing a homozygous recessive individual with a dominant parent, hence also called dominant back cross, because the individual, the dominant parent involved is pure homozygous tone. So what is the phenotype obtained here? All the tall plants actually obtained here are tall. So we can say all the plants, I can say either way, all plants obtained are tall plants. So it is not useful. So if you are getting all tall plants, then we can say now the individual involved, the dominant parent involved is homozygous dominant. You are getting the same result also in monohybrid. This is nothing but a monohybrid crossword. In a monohybrid cross, we are crossing pure breeding tall and pure breeding dog. We are getting the result all tall with the genotype capital T, small t. This is the genotype or obtained in F1 monohybrid cross. So we are writing in a different manner the same one. That is what is called homozygous tall test cross, as the dominant parent involved is homozygous. So these are the 2 squares giving explanation about, that is the back cross, recessive back cross, test cross, and we are taking what is called heterozygous tall test cross and homozygous tall test cross, otherwise called as a dominant back cross. and this is otherwise called as recessive backcross. So recessive backcross and dominant backcross based on the nature of the parent involved. Mendel took a pair of contrasting characters for each trait. Suppose we are taking for example tall and then dwarf. So these were the contrasting traits for the height. So logically we can say there are two alleles. One allele, capital T, another one, small t. This is the dominant allele and recessive allele. Now, during fertilization, we have just received, or Mendel received, what is called a hybrid tall. Hybrid tall. So this is what we have the pure parents. So it means that the recessive trait or its alleles do not disappear, but hidden or masked, only to reappear in the F1 generation as 1/4. In the F2, we are getting 3 tall and 1 dwarf. So, the recessive trait or its alleles do not disappear, but hidden or masked only to return back to get 1/4 of the population in the F2 generation. So, Mendel concluded that the dominant and the recessive alleles segregate randomly into gametes. He was the first scientist to use quantitative analysis to biological experiments. His data is concerned with the proportion of offspring. His data is concerned with the proportion of offspring. So this analytical, what is called approach, is generally, or we can say truly, an outstanding scientific work. His meticulous work, what is called giving great attention and precisely, that is, executed experiments. So his meticulous work, the great, what is called actually attention, and precisely executed case experiments propose that we have certain particular hereditary units present in the organism, which are being transmitted from one generation to another generation. Okay, and his experiments are normally just well planned. What for? To determine the relationships which govern the hereditary traits. Well planned to determine the relationships and which govern the hereditary traits. And this rationale, the board is given in the book, this rationale, what is called logical basis. This logical basis is called empirical approach. And the laws which are arrived at through empirical approach are called empirical laws. So this is a concept as given in the book regarding the analytical and empirical approach. So what approach he made? So what are the impacts of that approach? Okay. See, I mentioned about Mendel did experiments. His experiments normally concluded that actually the foundation for, he was the first person who has given foundation for, that is the inheritance, founded for the principle of inheritance, and revolutionized the field of biology. That's a major aspect of Mendel's work. So that's all about, we have the Mendel's analytical and empirical approach regarding his work, what for he did, in what way he approached both analysis and empirical formulae of the laws.